Let me start by introducing our first speaker today, who is Mark Chaplain. He is hiding back there. Mark is also a past president of the Society for Mathematical Biology. In fact, I will tell you that at this meeting we have the largest gathering ever of past presidents of the Society, which is really exciting. And on Wednesday, there's going to be a special award ceremony that we're hoping that you'll all come to and learn a little bit about the history of the Society and also the future directions of where we're going. So to start with Mark studies the growth of cancer and tumors in various ways and approaches. He was actually at the University of Dundee for a very long time. So what I mean by that is 1982 to 2015. So that's like 35 years, I think. And but finally he got sick of it, or they got sick of him, and he left. And he is now at the University of Andrews, where he holds the Gregory Chair of Applied Mathematics. And he'll probably stay there for 35 years. Mark has over 150 publications in math biology, and actually the Bulletin of Math Biology is the official journal of our society, and Mark claims to hold the record for the highest cited paper in the Bulletin, Continuous and Discrete Mathematical Models of Tumor-Induced Angiogenesis, and that was with Anderson. And it has 546 citations, but Alan and Reinhardt, you'll have to prove that right or wrong before the meeting's over, okay? Let's see, some other noteworthy things about Mark are that he was recently appointed co-editor-in-chief of the Journal of Theoretical Biology, and unfortunately he's also a Yankee fan, which I'll have to explain to you why that is, since he's from over the pond. And as I said, he was president of the society from 2005 to 2007. So without further ado, Mark, we'll welcome your talk.
Thank you. Okay. Thank you very much. Good morning. First of all, thanks for your invitation to speak. It's a pleasure to be here, and I'm looking forward to the week of talks here, and just to thank Fred and his organising team for all the hard work they've done so far, and what promises to be a really exciting meeting. And I wasn't quite in Dundee for 35 years. I did spend 6 years in Bath, down south in England, in the 90s, but it was a long time in Dundee, indeed. And for those of you who know your geography, St Andrews is just a short distance from Dundee, so I've hardly moved at all. So this morning I'm going to talk about some aspects of cancer modelling that we've been doing in our team over the last few years, and especially looking at cancer at different scales, which is in the title of the talk. So the overview is I'll give a brief introduction to biology, and then start focusing on the models at different scales, sub-cellular, cellular, and tissue scale, and then a bit of a roundup at the end, and then coffee. Okay. So before I get into the modelling, it's good to understand cancer, and I have a little anecdote to tell which will contextualize modeling and how to model a system, and it relates to attending a conference. So the story goes that there's 3 mathematicians and 3 biologists going to, let's say, a math biology conference, and the biologists are a bit better organized than the mathematicians, as always, and they arrive at the train station in good time. They buy their 3 tickets and wait for the train on the platform. The mathematicians, being a bit disorganized, arrive close to the wire. One of them goes into the ticket office and buys 1 ticket, arrives on the platform, and the train pulls in. Now, of course, there's a veneer of civility in these situations, but underneath there's a lot of competition. So the biologists smile at the mathematicians, go on the train into the carriage, and in due course, the ticket collector comes, takes the biologist's ticket. The biologists smile down to the mathematicians at the back of the carriage, and the mathematicians then leave the carriage and go into the toilet. The ticket collector comes down, knocks on the door. They have 1 ticket, ticket stamped, back under the toilet door, and the ticket collector carries on. The mathematicians return to the carriage and smile back at the biologist. So the conference comes and goes. Everybody has a good time, sort of, with the competition there. It's under the surface. Same story going back. The biologists are well organized. They arrive at the station, send one of them into the ticket office to buy 1 ticket, land on the platform. The mathematicians cut it really fine, straight onto the train without going to the ticket office at all. So they're sitting in the same carriage. The biologists smile back to the mathematicians. The ticket officer comes in. The biologists run to the toilet. the mathematicians get up, go to the toilet door, knock on the door, take the biologist's ticket, go into another toilet, and you can imagine what happens. The biologists get thrown off the train. And the mathematicians are very happy. But there's a moral to this story, and the moral is you should only model a system if you understand the system. So in order to model cancer, we should try and understand a bit about cancer. So when I started out in my PhD many, many years ago in the mid-80s, I went to a textbook of pathology, and although lots changed in cancer over the years, cancer itself hasn't changed. So this is a very good definition of cancer. It's a process where cells gain excessive proliferation and generate a lump. That's a solid tumor. You can classify tumors into two main categories. There's benign tumors, which essentially just grow as big lumps. In the talk, I'm going to focus on solid tumors. Of course, there are other tumors, blood cancers, brain tumors is a special class. So I'm going to focus on solid tumors, which are over 90% of all cancers. And then there's malignant tumors, which are called cancers. And the cancers are the ones that really do all the damage by invading and spreading and killing people, unfortunately. So if we get a little bit more focused, cancer is the disease of a cell. And you can examine a cancer cell and look at all the differences, the main differences between a cancer cell and a normal cell. And this is a photo from a paper I found when I was doing my PhD, and it lists very nicely all the main differences, starting at biomechanical differences between a cancer cell and a normal cell. Cytoskeletal components such as actin are modified, so a cancer cell feels different from a normal cell. Interactions with the surroundings through adhesion are changed. There's a response to the immune system that tries to fool the immune system. There's some growth factors secreted which allow the cells to proliferate even faster. There's an induction of blood vessels, and there's interaction with the local tissue, creating gaps and allowing the cells to proliferate into them. Now, this is a picture from a paper I discovered in the late 80s, and it's got very few citations. And it was summarized very elegantly in two recent papers by Hannan and Weinberg, and there's 14,000 citations of these papers each year. And these papers summarize that little cartoon into what Hannan and Weinberg called the 6 hallmarks of cancer, which is what I described there. Angiogenesis, the ability to generate blood vessels, the ability to invade local tissue and to metastasize, to spread into the surrounding body tissue and secondary sites. And then there are some hallmarks that are related at a cellular level or intracellular level. And all cancer cells have these hallmarks which distinguish them from normal cells, and of course lots of cells together form tissues, so a cancerous tissue has all these things going on. And Hannan and Weinberg were at pains to say that cancer is not really a genetic disease, it's a disease that you should really think of as interactions in the microenvironment with the cancer and the normal cells. So in terms of trying to assess cancers and make prognosis and work out if something's gone cancerous, the clinicians and pathologists have different ways of assessing whether something, tissue or cells have gone cancerous. This is at the basic level, the genetic level. So there's DNA screening, so you can see if there's mutations that have arisen in a cell line which indicate if things are going to go cancerous. And this is especially useful in certain cancers such as breast cancer. There's a known gene called BRCA1, which if that's mutated, there's a high probability you'll get breast cancer. And this is genetically related to your maternal line. So if your granny or your mother or your aunt has had breast cancer in this BRCA1 mutation, and you're tested and you have the mutation, there's a high probability you will have breast cancer as well. And if you can get that early, you can take action, remove the breast is an option, or treat early with chemotherapy or whatever. And so that's big strides have been made in treatment of breast cancer through genetic screening. So that's at the basic level of genes.
Mark,
hello.
That was discovered 3 blocks away.
So that was discovered 3 blocks away. Thank you, James. James will maybe fill you in on that a bit later. I didn't realize that. Thank you, James. So at the cell level, if a histopathologist gets some potentially cancerous cells and looks down a microscope, the pathologist can determine just how bad the cells are in terms of their shape, how they look, how they proliferate, and this is called grading. So you can give cancerous cells a grade. So low grade is better than high grade, but once a pathologist grades the cells, that indicates what you might do for treatment, and that's at the cell level. And then at the tissue level, pathologists can do what's called staging, and there are several options for staging, but a system that's widely used is called the TNM system, tumor, node, and metastasis. So the T aspect tells you how big the tumor is. The bigger it is, the worse it is. So there's a scale from 1 to 4. N tells you if it's spread locally to any lymph nodes, which isn't a good thing, because once you're in the lymph system, you can be transported around. And M is the metastatic stage, which is really bad. So either the cancer can remain localized, or it can be spread to other parts in the body. And if it's spread to other parts in the body, then that's really bad news and very difficult to treat. And it's the metastatic spread which really kills most people. So trying to keep cancers localized and small is the aim of a lot of treatment these days, rather than remove tumors at all. If you can live with a small tumor that's not doing very much. So the summary is that cancer, like most cellular systems, is a multiscale system. This is a piece of breast tissue that I obtained from colleagues at 9 Wells Hospital in Dundee. And you can see this is the normal tissue surrounding a breast cancer, which is a few centimeters in diameter. Close to the edge, if you focus in, here's the cancer normal tissue boundary. You can see it's very heterogeneous. Cancer cells are interacting with the local stroma and producing a very disparate and heterogeneous pattern of cells mixed in with normal tissue. And then at the cell level, there's lots of interesting things happening inside the cell, which are giving rise to these higher level phenomena. So, in terms of modeling, and the title of my talk, some years ago I took the time to read Turing's famous paper on pattern formation, and the opening sentence I think is really nice. The model is of a growing embryo, and Turing said, "This model will be a simplification, an idealization, and consequently a falsification of the system that he's studying." And I think that's a very elegant way of saying all models are wrong. But some models are less wrong than others, and even good models can make predictions and be helpful. But the process of modeling, as you're all aware, means you take a complicated system, make some assumptions, and simplify and work with some equations that you can get some results on. And if you've kept the features that are most important, then your simplified model can still be insightful for the overall system. So when I was a graduate student, there seemed to be an obsession in math biology with this phrase, and an obsession in trying to make every model into a system of 2 ODEs and do a phase plane analysis and bifurcation, and everybody was happy. Except that that was clearly an oversimplification in many cases. And in fact, I actually looked up who Occam was. He was a Franciscan friar in the Middle Ages, and he never said a model should be as simple as possible, but no simpler. What he did say in Latin was this. So I'm going to give you an exam. Best translation at the end of the talk will win a prize. No, it won't. This is what he actually said. He said that you shouldn't necessarily complicate things. You shouldn't complicate things unless it's absolutely necessary, which is maybe the same as saying a model should be as simple as possible, but I think philosophically it means if you try to make a simple model and it doesn't work, you should be open to making it slightly more complicated to try and connect with the real system. So if we come to a system which is too easy and it doesn't give the results, then we should be willing to make a better, more complicated system. The other thing that we have to, especially nowadays with all the data out there, this is a quote by Thomas Huxley, and again I think this reminds me of my early days in math biology. People used to make models just because they were a nice set of equations which had an elegant solution. If they didn't match what they were meant to model, nobody really cared because they had a nice solution. That was good for the mathematics, but it wasn't good for the biology. So I think we really need to remain faithful to the data, and our models, at the very least, must replicate what's already known. But that in itself isn't very good because the biologists already know that, so we have to go a bit further and be predictive and quantitative in our results. So, on to the actual modeling. First of all, at the intracellular level, this is from Hanahan and Weinberg's paper. This is some key pathways inside any cancer cell. So you can see there's a lot going on, and this is just a subset of what's going on. And I was interested in intracellular modeling because of two molecules in cancer, p53, which controls apoptosis, and NF-kappa B, which controls various responses to cells which can lead to better treatment. And in particular, p53, that's a huge molecule in cancer research, and for a long time people thought if they could get a drug which acted on the p53 system, you would literally cure all cancers. Of course, it's a bit more complicated than that, and the breakthrough in drug treatment has not quite appeared. So I was at a talk when somebody was giving, I was at a conference when somebody gave a talk on NF-kappa B, and they showed this movie. I was really intrigued. Because up until that point, I hadn't really modeled anything inside the cell. I'll just show that again. So the NF-kappa B here has been stained with red fluorescent protein, and these are single cell images, and what you can see, the nucleus lights up and goes dark, lights up and goes dark. That's because NF-kappa B is a transcription factor. Transcription factors are proteins that bind to DNA, and if you bind to DNA, then you have an effect on RNA, and these transcription factors can upregulate or downregulate the production of mRNA. And of course, if you do that to mRNA, then you've got a knock-on effect to whatever protein the mRNA is making. Okay, so these form a part of the larger class of what's called gene regulatory networks. P53 and NF-kappa B are both transcription factors, gene regulatory networks. And they are negative feedback molecules. So what that means is that this is a generic negative feedback loop. A molecule X, and this arrow could have several intermediate pathways. Molecule X eventually makes a molecule Y, and molecule Y, either directly or indirectly, feeds back and shuts down the production of X. And you can just argue heuristically that if you have this negative feedback system, you get oscillations in the molecular concentrations of the species. Now, there is a simple feedback system of one feedback loop of the protein feeding back onto the mRNA, and it's called HES1. And this is some data from 2002, Japanese group. They studied this protein. It's got this molecule. It's got implications for differentiation of stem cells. You can see very nicely from the data that this oscillates with a period of 2 hours because of the feedback loop. The protein shuts down the mRNA, and the mRNA then makes less protein, but then the mRNA can rise again, and so you get these oscillations. So 2002, there's a lot of systems biology going on, so people started looking at this system. Because it's so easy, you can write down a couple of ODEs if you assume your cell is a well-stirred chemical reactor. So here's our mRNA. It's made at a rate alpha m, and the role of mRNA is to make protein. So you make some protein. The protein and the mRNA just decay, and then the protein feeds back and shuts down the mRNA. So you can put in your favorite feedback function f and see what the outcome of the model is. And the outcome of the model is that you don't get oscillations for any feedback function f. So what people started to do was they put some delays in, and if you put a delay in, you're bound to get some oscillations because that's what delays do. So I had read the literature and realized that this was available, but I was unconvinced of the argument about delays, and this is essentially just a single compartment model, and there's a nucleus and a cytoplasm, and the molecules are in different compartments. So I decided to make a spatial model, partial differential equation, a little mathematical cell with a nucleus, a cytoplasm, a nuclear membrane, and a cell membrane, and we just let the molecules diffuse and produce and decay in the correct compartments. So here's the PDE model. The mRNA is produced in the nucleus, so subscript M is in the nucleus, subscript C is the cytoplasm. So it's produced in the nucleus, it diffuses, goes into the cytoplasm, makes some protein in the cytoplasm, and then the protein diffuses back into the nucleus and downregulates the mRNA. So everything is just the same reaction kinetics, but in the correct compartments, and there's diffusion. And you've got some boundary conditions on the nuclear membrane, which you can modify as you want. And you get very nice oscillations with the system with no delay. So the spatial movement of the molecules is very important. And by changing the diffusion coefficients just numerically, we noticed that if you made the diffusion too big, you lost the oscillations. If you made the diffusion too small, you also lost the oscillations, which kind of indicated some Hopf bifurcation. And recently, with a colleague, Maria Potashnik, in Dundee, we managed to prove rigorously, well, Maria did all the hard work, Maria proved rigorously, and I could have an hour-long talk on this paper alone, so I won't bore you with the details, other than to say that the diffusion coefficient of the molecule is a Hopf bifurcation parameter. So space influences time. So it's the movement of the molecules that causes the oscillation. Now, you may argue that the movement is a delay, but I think if that's the case, you're better modeling it explicitly with a PDE rather than a discrete delay ODE. Okay, so we were pleased we could get oscillations without delay. But if you look at the real data is a bit stochastic, and the numbers of protein molecules in the system is relatively low, a few hundred. So you really shouldn't be using a continuum approach. You should be using a stochastic system. So we decided to make a, well, this is just the, so the data is taken from some experiments on mice embryonic stem cells. These are the oscillations of Hess1 protein in 5 separate cells over a period of a few hours. And you can see the sort of stochastic oscillations. And you can extract from those stochastic oscillations an average period. And this is the result of analyzing 100 cells. So you can see you've got quite a wide variety in the length of the period. So we decided to go back and make a spatial stochastic model of this system and implement a spatial Gillespie approach. So you get a master equation with all your reactions happening stochastically, and you just treat diffusion as a reaction. So it's a very simple approach, and there's lots of software out there to solve these systems. In particular, we used a piece of software called URDME, which means unstructured mesh reaction diffusion master equation. So it's a combination of the Gillespie algorithm with a finite element discretization of your domain. I can go into the details over coffee if anybody likes to ask me, but I'll just get on to the output. So when you solve the system, our Hess1 system, stochastically in the spatial domain, these were the results. So these are results of 5 simulations, and as you can see, in the eyeball norm, it's pretty good. You can match these and convince yourself that you've got a good match. Before I get into a bit more quantitative assessment, you'll see here there's a green line, and that's at the time that the stem cells decide to differentiate. They differentiate in 1 or 2 pathways. And the differentiation into which pathways is determined by the number of molecules. And so you can see at this time, for example, here there's 620 protein molecules of Hess1. This one is 891, so that's a higher level. This one we're back down to 600, 957, and so on. So at the time of differentiation, you've either got high or low Hess1, so it's really just random whether you differentiate one way or the other. So there's no secret internal mechanism controlling differentiation. It's just down to the luck of the draw what the level of molecule is at the time of differentiation. And if you're a bit more accurate and look at and try and see if the periods really match, these are the simulations of 100 different simulations. So you can see you get a broad spectrum of period lengths, and that matches very nicely with the real data. So not just in the eyeball norm, but this is a proper analysis of the data. And we've done it with P53, and that also matches very well. And then back to where I started with NF kappa B, it's a big complex system. There's about 35 key reactions, and there they all are. That's all you want to see. And this is our simulations of the NF kappa B system. And as you can see, very nicely, we're capturing the molecules going into the nucleus and then going out of the nucleus, going into the nucleus, going out of the nucleus, just as it does in the real system. And again, when you analyze this properly, you get a very nice match quantitatively with the real data. But it's solving 30-odd reaction, diffusion, stochastic equations, which is quite a complex system. It's not just a couple of ODEs and a bifurcation. Okay, so moving up one level to cell-level modeling. So a different approach. And back to our little cancer cell, which is different from a normal cell. And one of the key differences is in adhesive properties. So cancer cells stick to each other differently than normal cells do. They have reduced adhesion to each other and a bigger adhesion to the local matrix. So in order to do that, we've developed some individual-based, force-based models. This is based on work by Dirk Drasdo, who's been working in this area for many years and developed this approach, really. And now, when I started doing this 10, 15 years ago, there weren't many models out there of individual-based, force-based nature, but now there are lots others to choose from. So this is not the only way to do it. There's CompuCell, which is lovely. James Glazier's here in the front row, who made CompuCell. Paul Macklin's here in the front row, who's developed his own individual-based force model. So there are lots of nice ways you can model individual cells interacting, trying to get some insight from a biomechanical force-based perspective. And the underlying modeling assumption is just Newton's second law of motion. So force is acting on a body, producing acceleration. But if you're in a cell environment and cells move rather slowly, you can ignore acceleration. And if they're moving on tissue and substrate, there's a drag force associated with that, and you can assume the drag force is proportional to the speed of the cell, and so you get, for any given number of cells, you just get a large system of ODEs. So dx by dt equals the sum of the forces, and if you solve that, you'd work out where the positions of the cell are and how that changes over time. So conceptually, it's really quite straightforward. It's just a big computational mess. It takes a lot of time and effort on the computer. So the approach considers cells as little three-dimensional spheres. They have a biomechanical property associated with them, so there's some attempt to model them as little biomechanical realities. You allow the cells to grow and expand in the radius, and when they reach a certain radius, they divide into two daughter cells with conservation of mass. And you can give the cell division rate as to whatever cell line you're looking at. Fast-growing cancer cells divide about once every 24 hours. So if you put a bit of variation on your 24 hours, you can have a kind of stochastic division time lasting between 17 and 27 hours. So we have little spherical masses, and we assume there's some kind of force between the cells, which can be modeled by a potential function. And if you take the derivative of the potential, you get a force, and that is a force of repulsion, but there's also adhesive forces. And if you implement the potential function via the Hertz model, which is used in solid mechanics, the potential between two cells is given by this formulation. You can add in the adhesive forces between the cells, and there's the elasticity of the cells there. So that can vary between cancer cell lines and normal cell lines. And from that potential, this is the qualitative shape of the Hertz function, you get a force, and for two individual cells, you would get some kind of equilibrium where the forces of repulsion, if they're too close, balance the forces of adhesion, and the two cells would stick together. So if you have more cells, you just add up all the forces and solve your ODEs, and the cells would move in space, or they could reach some kind of homeostatic level. And the overall movement of a complex cellular system is just governed by all the forces, and so you just, in addition to adhesive forces between two cells, you have some frictional force between the cells and the external medium that they're embedded in. You have local frictional forces between the cells. That's the forces of attraction and repulsion. You've got a bit of noise in the system because there's always noise there. And then you can impose external fields such as chemotactic gradients and haptotactic gradients in terms of the density of the extracellular matrix. And as I say, Dirk Drasno formulated this approach in order to model epithelial sheets of cells or morphogenetic buckling in cells of little rings of cells where the bifurcation gave you an invagination, and that led to gastrulation and other important events in early development of biology. So, but up until recently, the interaction with the external medium was just done by a frictional force, which is a very big simplification of actual reality. So, a few years ago with a PhD student, we decided to, a PhD student, Daniela Schluter, we decided to model the interactions between the cells and the matrix explicitly by creating little matrix fibers, sort of collagen-like fibers, which were also biomechanical objects, rigid little cylinders. And so if the cells interact with the little rigid cylinders, they would pull on them and act like a lever in a fulcrum and turn a cylinder through a certain, excuse me, a certain angle. So if you have one fiber and one cell, the cell would attach with a given force, pull the fiber through a certain angle, and realign the fiber. And then use that aligned fiber as a direction cue to move in a given direction. If you interact with multiple fibers, you move all the fibers you're in contact with, and then you take an average distance in which to then start moving in a given direction. So again, it's just a computational exercise in counting the fibers in contact with the cell, moving the fibers, working at the average new direction, and carrying on in that direction. So here's our little mathematical cell, embed in a two-dimensional domain of fibers, and you give it a little push, and it interacts with the fibers. It realigns the fibers and uses those realigned fibers to move and migrate on. And as you can see, you get behind the cell track, behind the path of the cell, you get these realigned fibers which have allowed the cell to move in a given direction or to change direction. And you could examine how the matrix density, how the alignment of the fibers.
Mark, is there stochasticity there?
Yes, the noise force, the noise bit gives you stochasticity, so that's a bit of randomness, exactly. So let me just. So you can do that with two cells once your one cell is working, and they kind of play follow my leader. So the lead cell rearranges the fibers, and the second cell sometimes follows it, it sometimes doesn't. So that's proof of concept again. And then you can do lots of cells, and they have an interacting time. You can see they're really remodeling the fibers here, and so where the fibers are really remodeled extensively, you get empty space, and then the cells can't move in that empty space. So that has some implications for cancer invasion, epithelial mesenchymal transition, and cells need to interact with matrix fibers to spread locally. But at this stage, it's just a little computational model showing that we can interact cells with fibers and move them. So again, to try and then test the model precisely with a real experiment, once you've got your basic model going, we hunted in the literature and found this nice paper from 2000 from a group in Boston, Mike Adembo's lab. And what they'd done was they created a matrigel half and half. Half of the matrigel was soft, half of the matrigel was stiff with more density of collagen fibers in it. And what they did was they placed a single cell, a fibroblast, on the border of the transition between soft and stiff. And they put the cell either on the soft side and then came back some time later and worked out where it ended up, or they put the cell on the stiff side and again came back later and worked out where the cell ended up. And they found that on average, if you put the cell on the soft side, it liked to go to the stiff side. If you start on the stiff side, it prefers to stay on the stiff side. So we made our little mathematical petri dish with a soft side and a stiff side. We put our cell in the middle on one or the other side. We ran the simulations for the appropriate length of time, and asked where our little cell ended up. And our results showed the following. If you start on the soft side, these are 15 simulation runs, so the vast majority of the time you always end up on the stiff side. If you start on the stiff side, you always remain there. Now, the description, the explanation of Dembo's lab for this phenomenon was that there was a lot of intracellular signaling with actin and myosin and all sorts of everything in the kitchen sink was contributing to a complicated intracellular machinery which was allowing the cell to move to the stiff tissue. Whereas our little simulation seemed to indicate that a simple biomechanical aspect, that the cells just prefer to stick to the hard tissue, and there's no intracellular machinery explanation needed. So that's a simpler explanation for the experimental data, which generally speaking is always is to be preferred. And you can carry on and do investigations of how fiber density, how fiber alignment affects the individual cell movement, and unsurprisingly, all these things are important. The matrix stiffness controls the cell speed. Matrix alignment is important. If the fibers are very rigidly aligned, then the cell has to basically follow the fiber if it can't align them. And you get interesting relationships between the persistence times of the cells. The persistence time is how long a cell keeps going in a given direction before it changes. So all these things have an influence on persistent times and how cells move through matrices. So the model is producing quantitative predictions of cell matrix interactions. Now, of course, it's always more complicated than you want. And what's going on inside a cell actually is important in terms of adhesion. This is a little movie of epithelial cells. And when epithelial cells meet with other epithelial cells, you get two proteins called E-catenin and beta-catenin move to the membrane and make adhesion bonds between them. So the intracellular levels of these two proteins, E-catenin and beta-catenin, are really important in the adhesive process. So and this is what's happening. E-catenin and beta-catenin are normally in the cytoplasm, but when two cells contact, the two molecules are brought to the membrane where they form a complex, E-catenin and beta-catenin complex, and those complexes allow bonds of adhesion to form between the two membranes. If you pull cells apart, the molecules are released and then they go back into the cytoplasm. So you can use a little law of mass action. You can write down a system of ODE equations governing the levels of E-catenin, beta-catenin, and the complex inside each cell. And so now every little cell has a system of ODEs governing the adhesive forces between other cells. So it's even more computationally expensive. But you can answer some nice questions. There's a hypothesis experimentally as to how the E-catenin and beta-catenin join not just two cells, but multiple cells when they're bound. There's what's called a static adhesion hypothesis. So that means when the bonds are made, they're made and they're really fixed and they can't move. And if another cell comes along, you have to make new bonds and stick the second cell together. The dynamic hypothesis says if you make two cells connect and another cell comes along, you can redistribute some of those bonds to the new cell that's connected. It's a bit more efficient, and that's called dynamic adhesion. So we ran our model under both assumptions. There's some data just looking at the forces between two cells and multiple cells over time. So the experimental curve is the dotted curve. And when you assume a static adhesion hypothesis, the model predicts the force over time as constant, which doesn't fit the data at all. But if you assume the dynamic adhesion and you have the rearrangement of the molecules, then the model predictions match the data really quite well. So it seems to indicate that dynamic adhesion is what is controlling multiple cell connections. So in summary, we've now got a very nice individual-based model, which is genuinely multiscale. We have intracellular signaling controlling cell-cell adhesion, and if you simulate enough cells together, you can get to a kind of tissue level, which is the next part of the talk, which I'll get through as quickly as I can because it's nearly coffee. 129 slides. Oh my God. I'll tell you about that in a minute. So I don't have 129 slides. So tissue level modeling. This is some real data from real cancers and real people from a Japanese group. This is esophageal cancer. And you can see here there's some staining, and we have what's called an infiltrative invasive pattern A. And this is good because the cancer has been localized. There's a clear boundary between the cancer and the normal tissue. And then at the other end of the scale, we have infiltrative pattern C, which is a complete mess and is not good. So it's a very poor prognosis of survival with infiltrative pattern C. So if a pathologist looks at this tissue biopsy from a patient and you've got infiltrative pattern C, it's not good, and you have to get treatment quickly, or if it's left, you'll die quickly. There's something in the middle which they just said was neither A nor B. So you have two extremes and something kind of in the middle. So this is real data from tissue level analysis of cancer invasion. This is related to a phenomenon called haptotaxis, where cells adhere to the matrix, and there's differences in matrix density allow the cells to migrate in a given direction. Mathematically, it's the same as chemotaxis, except the molecules here don't diffuse. So if you derive a model at the tissue level, it's natural to use PDEs. And so we have the following key interactions going on. The cancer cells secrete some enzymes called matrix-degrading enzymes. They degrade the tissue. The tissue is then changed in density, and the cancer cells can use the change in density to move in a directed manner via haptotaxis. So you get directed invasion. And if you just write down the standard kind of PDE that you would write down phenomenologically in this case, you would use a reaction diffusion system, a bit like Fisher's equation, with a haptotaxis term in here, which allows the cells to invade. You simulate this, you get traveling waves, as I'm sure people who are familiar with reaction diffusion systems know. And so the model is confirmatory, but once you give initial conditions, it just keeps invading, which is not what all cancers do. And the reason that the model is not particularly good at describing all invasion is that there's no cell-cell adhesion in this model. There's cell matrix adhesion, but no cell-cell adhesion. And that's because cell-cell adhesion in a PDE model is very difficult to do, just with the usual tools. And so some years ago, we borrowed a very nice idea from Jonathan Sherritt and Kevin Painter, who were looking at cell adhesion in an embryogenesis scenario. And they developed a concept of an adhesive flux between cells. And this is essentially the same model system I gave in the last slide, but there's a modified term here which models cell-cell and cell matrix interaction. And the term is a non-local term, so there's a spatial kernel. There's what's called a sensing radius, because cells like to sense their local environment. So for some sensing radius R on the level of a single cell, cells can explore their environment, and they can interact with the cancer cells themselves, so that's like self-adhesion, and they can interact with the matrix, so that's standard haptotaxis. And in fact, you can show, if you take this system of equations and let the sensing radius tend to 0, you recover the standard haptotaxis PDE you would write down under normal circumstances. So it's a generalized haptotaxis system. And we can add in a bit of mutation. I'm just going to skip through a little bit here. We can use the knowledge of cancer aggressiveness, where over time cancers become less adhesive to themselves, more adhesive to the matrix, and allow a time-dependent adhesive parameters in the model, and then just simulate the system and see what happens. So these are our cancer cells, the matrix, and some initial matrix degrading enzymes. And as you change these adhesive parameters, we got some very nice spatiotemporal heterogeneity appearing quite naturally. So first of all, we were pleased, just from a mathematical point of view, because we get nice patterns. But when we connect back to the real data, that really shows that the loss of cell-cell adhesion and the gain in cell matrix adhesion may be the real the key underlying thing describing those two extreme infiltrative patterns. If you have strong cell-cell adhesion, you're in infiltrative pattern A. If you have strong matrix adhesion, you're in infiltrative pattern C. There's also some in vitro data. This is an organotypic assay. So you put cancer cells on top of collagen gel, watch them invade, and you can mutate the system by knocking out collagen, and we can do that in our model with the non-local equations. This is the baseline simulation, and then if you knock out the adhesion parameter, you get this more extensive spread, which at least qualitatively in this case matches the real data. Okay, so some conclusions. So for me, personally, like Captain Kirk said, space is the final frontier. Space and how individuals interact in space is really important. And this also raises its head in other models we've done, especially in chemotherapy drug treatment models. If you deliver drug to a tissue, the drug diffuses and decays. And so if you're a cell, your spatial location really matters because you're not getting the same level of drug. The level of drug you get will depend on your spatial location, and it becomes very nonlinear and complex very quickly. So space is very important. Also, connecting the different scales is very important because the scales influence each other both bottom up and top down. So I think we really do need to try and keep focused on multi-scale models. You also have to be willing to be flexible in your modeling. If you have a PET modeling approach, you can't use that approach for all circumstances, so I think you have to be willing to forget your PET approach and derive a new approach if necessary. And not only is good modeling predictive, but as a little aside, which is a positive thing, you sometimes develop new tools, for example, numerical methods. The non-local model that we had, my colleague Alf Gerish, he had to develop an efficient computational tool to solve these systems. If you just take a very simplified, naive discretization approach, it takes forever to simulate. So he developed a very nice, fast, new technique, which he was able to publish in a bona fide numerical analysis journal. So good modeling leads to good things in other fields as well. So just last couple of slides on future perspectives. If we go back to our bit of breast cancer, at about a centimeter or so, there are about 1 billion cells in that piece of cancerous tissue. That's a lot of cells. And so you'd be tempted to say, well, we'll have to use a PDE model because simulating 1 billion individual cells with all the forces, etc., is just beyond my desktop Mac. But if you have colleagues who have access to a big, fast parallel machine, which is in Warsaw, then you can simulate. This is a real simulation of 10 to the 9 cancer cells. And it takes about 2.5 days. Now, normally this would take 6, 7, 9 months to grow in vitro. So we're now at the stage of being able to solve 1 billion interacting cells. And of course, hopefully you've seen that mutations and differences in small numbers of individual cells can really have an important effect at the population level. And at a PDE approach, you can tell what's happening at a population level, but it's very difficult with PDEs to work out what's happening with single individual cells. So I think we're now at the stage where we can do large-scale computation and try and connect large-scale computation with PDE models and get the best of both worlds. So that's the future for me, trying to create a virtual tumor, multi-scale simulations, and connecting with all the stuff in PDEs that we know and love and can do nicely with PDEs. And just a bit of plug, not for me, but there's a new issue of the Bulletin of Math Biology just out. It's devoted on mathematical oncology. It's edited by Philip Maney and Sandy Anderson. So that's some of the articles are on. Philip, is the issue actually published, or is it just online?
It's online.
Yes. There are articles online, and then once we've got enough articles online, there'll be the special issue with page numbers and all those kind of things. And if anybody, I know all you young people are nice, you like to tweet and things, so if you are going to tweet, I personally, a personal plea, I just found out this morning that Sandy Anderson, who's a very good colleague, worked with me a long time, he, ironically, is in Dundee this week. So if you're going to tweet, could you please tell Sandy why isn't he here? And he's missing a great conference. I'll leave the last slide to thank all my collaborators. Thank you for your attention, and I look forward to interacting with you over the rest of the week. Thank you.
What you might can do is move to the end of the aisle. If you want to ask a question, just we don't have to climb over you.
Yeah, thank you very much. Very nice talk. I like your mathematical result that so spatial structure diffusion can induce oscillation. So even in the two variable negative feedback system. But in biology, I think so Hess-1 oscillation is multicellular behavior, and it is strongly suggested that cellular interaction is so very important.
Yeah.
So I think some idea. So you have some idea also why you omit so spatial structure in your.
Why I've omitted spatial
Yeah.
structure?
So because also there are so many also mathematical. So I think both mathematical and experimental studies which suggest cellular interaction. Coupling is very important.
Okay.
Yeah.
Well, we focused on single cells in this case because
Okay.
the data was on single cells. Okay.
Okay.
I agree. If you want to model a collection of cells all having Hess1 signaling, you have to then have cell-cell interaction and communication. But we've modeled a similar situation in somatogenesis. There's a HER7 protein, Notch delta, etc., and that controls oscillations in adjacent cells. And you can block things on the membrane, and the systems are asynchronous. And if you open up the membrane and Notch delta is active, you can synchronize the two cells. So we've looked into that a little bit. Yeah.
So in previous studies, spatial interactions are strongly suggested, but also oscillations are observed even in single cell.
Yeah.
Okay, I see.
So yeah, I have a question about, in the case of cells are interacting each other, the governing equation turns out to be diffusion reaction, reaction diffusion equation. So what you mentioned is, I think, reaction diffusion equation has a traveling wave form solution, but the example, the picture you showed us seems not have any pattern. So that's my curiosity.
Okay. Yeah. So the traveling wave occurs when you have the standard PDE without the non-local interactions. You just get traveling waves. But if you include the non-local interactions, you can still get some traveling waves in certain cases, in certain parameter regimes. But what the key to the model, the results that I showed, was we had the adhesion parameter was time dependent. So the adhesion parameter changed over time, and that gave rise to those interesting heterogeneous solutions. But if you have a fixed set of parameters, even with cell-cell adhesion, you can get a traveling wave solution. It's still there. It's just this gives you a wider set of class of results.
Anybody? So I have a question for you, Mark. So we talked a lot about multiscale, and at the very end you kind of hinted at this, but are there models, or do you think it's important to connect the models, even if they are hybrid and they're all of a different flavor? You kind of said at the end, well, you can have a stochastic discrete model that maybe has some PDE diffusion or something in it, but what about connecting north-south, kind of? Is there from a cellular to molecular all the way up?
I mean, I think we did that a little bit with the individual cell-based model, where we had systems of ODEs inside each cell. So the ODEs were modeling EGADR and beta-catenin levels inside each cell. We had to make the assumption there that it was a well-mixed chemical reactor and there was no nucleus in cytoplasm, because I think solving a PDE inside each cell and trying to solve 1 billion interacting cells with systems of PDEs inside them is beyond even a supercomputer. So you still have to make certain simplifying assumptions there, but I think having systems of ODEs is better than just a constant, for example, and you can cope with ODEs. And the other thing is that I don't want to give the impression that you have to model everything inside a cell with a PDE, because the transcription factors are a special case because the protein has to go into the nucleus. So the two compartments in that situation are very important. Other situations you can just model levels of proteins with ODEs because there's no feedback into the nucleus.
And it also may depend on the question because you may not really care about the
Absolutely, yes,
location,
that's
so you can
very
coarse
important.
grain it.
Yeah,
So
you know, there's no point in deriving a huge complicated model if you can get it, if the question that's being asked of you, you can do it in a simple way, yeah.
And then one other thing, so you sort of talked about two systems, the breast system and then it looks like the gut system there, or the mucus surface. So do you find that there are some lessons that are applicable in both kind of settings that are operating, or are they very different just because the environments themselves are so different?
So we've taken the approach to try and develop a generic model of cancer interacting with tissue. If you wanted to try and focus that on a specific breast cancer or liver cancer, you would have then to be a bit more specialized in parameter values or the type of cells that you have in the local environment. But I mean, certain cancers, certain locations in the body, for example, the gut and crypts and all that have very specific geometries which, you know, this type of modeling isn't really appropriate for. But for carcinomas in general, this gives a lot of insight without having to specialize.
Anyone else have any questions? Okay, well, let's thank our first speaker again this morning.